What's up, BloodStream listeners, and welcome back to another episode of Gene Therapy, A Whole New World. It's your girl, Hannah, and today we are traveling through time to explore the history of gene therapy. From the first sparks of discovery to the major milestones that brought us here, FDA approved gene therapy for hemophilia, and how hemophilia helps prove what was once thought to be impossible. And to help us tell this story, we're leaning on Portraits of Progress, found at www.portraitsofprogress.com, a fun initiative by CSL Behring celebrating the community's amazing journey and progress along the way. It maps it all out, from early plasma treatments in the 1940s to the first gene therapy experiments in the 80s, all the way to the first successful hemophilia trials in the 2000s. Joining me today are voices from every part of that timeline. Dr. Mark Kay from Stanford University, a pioneer whose work helped make those early trials possible. And Ying Poi Liu, a scientist bridging research and community. Dan and Jerome, community advocates featured on Portraits of Progress, sharing their experience living with hemophilia. And of course, Greg, whose perspective as a patient who's actually received gene therapy brings this story full circle. Though before we jump in, here's a quick word from our sponsor. It's a Whole New World gene therapy segment brought to you by CSL Behring and their Hemophilia B gene therapy treatment. To learn more about gene therapy, hear patient stories, and view additional support resources, visit beyondhemb.com or download B Support wherever you get your apps for more information and to manage your treatment journey. Now, let's head back to the early 1980s when the dream of gene therapy was beginning to take shape.
The cloning of the factor 9 gene and the factor 8 genes in the early 1980s was a major breakthrough in science. This was, of course, essential in order to develop gene therapy, because in gene therapy we use the defective gene in order to deliver that to the patient via vector. And so the patient will eventually, after treatment, produce its own clotting factor, and so no longer need to have infusions anymore.
That discovery, cloning the very genes responsible for hemophilia, well, it gave scientists the map they needed to start dreaming about genetic cures. But back then, this was still brand new territory. No one really knew if it could work. And for many families, the 1980s also carried deep heartbreak.
Unfortunately, in the early 1980s, many people with hemophilia were infected with HIV and HCV viruses due to infusions with infected blood. This tragedy pushed the scientific community even harder to develop new and safer treatment options.
Out of that tragedy came determination. Researchers doubled down, and some of those scientists, like Dr. Mark Kay, would spend decades chasing the dreams of a safer, longer-lasting solution.
I've worked in the gene therapy field since the early 1990s. I think when I started in the field, the concept of gene therapy was relatively simple, and there were a lot of promises made that weren't fulfilled, because it turns out there were a lot of technical barriers, some of which at the time it wasn't clear, obvious how to try to study that to get around some of these barriers. But over time, over the last few decades, a number of achievements have been made. We made contributions, many others, that have really pushed the field forward, where it's become something that has gained a lot of respect.
Dr. Mark Kay would go on to lead some of the earliest gene therapy research in hemophilia. But to understand why hemophilia became such a central test case, you have to start with the people who lived through those early years. So on the Portraits of Progress timeline, this era marks the beginning, the 1950s and 60s, when treatment was limited, risky, and often experimental.
I had a pretty significant history of hemophilia in my family, so they were ready for it. They had in the delivery room to give me a direct transfusion if I needed it. I didn't, fortunately, but they were prepared. So I was diagnosed, as soon after birth as possible, back in 1953. So I was kind of on the leading edge of hemophilia research when I was 5. And I was subject to numerous experiments. As a child, I remember a great deal of going in the hospital, because that's the way it was treated back then. There was no outpatient, there was no home therapy. So every time I had to bleed, I had to go into the hospital. And then it took a week or more in the hospital to resolve the bleed before they would release me. So I was in the hospital about a third of my life by the time I was 12.
Dan's experience wasn't unique. In those early decades, treatment was long, unpredictable, and as we heard from Ying, not always safe.
I was born before there was a diagnosis for factor IX. I had a younger brother who preceded me by a couple of years, who died at the age of 10 months with the disease, undiagnosed. So as a result of that, Dr. Quick from the University of Chicago was involved at my birth, and he did diagnose me within a few days of my birth that I was a factor IX Christmas disease. He had just weeks before diagnosed Stephen Christmas, and who the disease is named after.
Greg's story shows us just how recent this all is. Within a single lifetime, we've gone from no diagnosis to genetic discovery to curative trials. And back in the lab, researchers like Dr. Mark Kay were figuring out how to turn those genetic discoveries into real treatments, marking one of the biggest turning points you'll see on the Portraits of Progress timeline.
The real technology advancements were in the vectors that are the vehicles we use to transfer genes into tissues to try to treat genetic and now even acquired diseases. So as gene therapists, what we tried to do was to remove the viral genetic information, put in our therapeutic genes of interest, and use the virus shell or the outside of the virus to actually allow that genetic information to be transferred into the tissues that you wanted to target to treat a specific genetic disorder.
Those viral shells became the backbone of modern gene therapy. Scientists tested them in mice, then in dogs, and eventually in the first human trials. And one of those early volunteers was Dan.
I was in a gene therapy trial about 20 years ago. There was a safety trial where the goal was to make sure that they could do this without killing people. And they were successful. But the therapy itself didn't last. It only produced factor for about 6 months. But the thing is that there's a direct line between the trial that I did back then and the trials that are going on now.
A direct line from early experiments like Dan's to the therapies now on the verge of changing hemophilia treatment forever. If you scroll through Portraits of Progress, you can see the evolution mapped out across decades, from those early safety trials in the early 2000s to the first FDA approvals that followed years later.
As a scientist, my hope for the hemophilia community will be that there will be many more different treatment options available for them, such as gene therapy, which can be a potentially curative treatment, so that they can pick and choose what's suited for them. And most importantly, I hope that all patients can have access to these transformative treatments.
That dream, access for all, is what drives so many in this field. And as Dr. Mark Kay explains, the discoveries that began in hemophilia labs are now reaching far beyond.
There are some trials that are going to start that potentially can get around that problem. And that is a way to take the AAV DNA that encodes the factor IX sequence and integrate it into a safe spot in the genome, in the hepatocytes. And there's a trial that I believe is going to start very soon, where the way they're going to do this is they're going to use a gene editing technology in addition to the gene therapy. So I'm sure that many of the listeners have heard of CRISPR or CRISPR engineering, but I think this is a hugely expanding space, and I think something that it's very exciting.
CRISPR and the AAVs, these are the words of modern medicine. But their roots trace back to people like Greg and Dan, to scientists like Dr. Kay and Dr. Liu, and to share a hope that one day a bleeding disorder wouldn't define a life.
I think gene therapy will do wonders for the community at large because it depends on the person and it depends on what their severity level is. It also depends on whether if the gene therapy is right for them as well, because most people have joint issues, and doing gene therapy will probably help them. It will help them not have that joint issues no more. It'll help them live a better life, and they can still continue to be active in their personal lives and their professional lives.
For Jerome, gene therapy represents hope, a new chapter waiting to be written. For Greg, that chapter is already being lived.
It was the infusion of Hemgenex. It's a treatment that I did not really expect to see become available in my lifetime. It just seemed like something out of a dream. The doctors fully understood what they were doing. I realized what almost a miracle this was, and the fact that it works so well. Just literally something that I never expected in my lifetime to get a chance to enjoy. I was just so fortunate at the age of 72 at the time to get a chance to experience this.
If you're visiting Portraits of Progress, you've seen faces like Dan's and Jerome's, each story showing how far we've come and how every step in research and lived experience builds us towards something bigger. And as we look ahead, the story is not finished. Next time, we'll explore what's still unknown, the future of gene therapy, and the questions researchers and patients are daring to ask next. From the lab bench to lived experience, that's the story of gene therapy, and it's still being written. If you've enjoyed today's episode, share it with someone who loves a little science history and a lot of hope. Thanks for listening to BloodStream and another episode of Gene Therapy, A Whole New World. Until then, it's your girl Hannah, and we'll see you next time.